So let's start today. Hello, everyone, and welcome to this member-to-member session, the last before summer holidays. And happy to have you all here in this Zoom session. My name is Stephanie. I'm the PR manager at bioRN, and I'm excited to highlight one of our member companies that help transform scientific ideas into real market-ready therapies, Probio. So Probio stands at the forefront of next-generation biologics development, specializing in the toughest challenges, bispecific and multispecific antibodies, antibody-drug conjugates, plasmids, and viral vectors, and supported over 90 CMC, CMO projects, and helped secure more than 30 IND approvals. However, they're not just a contract manufacturer, but also innovators. And I'm happy to speak today with Ying Li, to dive deeper into how Probio turns lab stage breakthroughs into clinic-ready biologics. So let's get started. Ying, let's start with you. Please tell us a bit about yourself and how did you personally become part of Probio?
All right. First of all, thank you, Stephanie, for hosting this session with me. And hello, everyone. My name is Ying Li, Director of Biologics Process Development at Probio. So my journey into bioprocessing began with my PhD study with Professor Andrew Zidney from the Pennsylvania State University, focusing on the purification of plasmid DNA using an innovative membrane-based process. I then spent 5 years at Merck KGaA in their US Boston site, where I managed platform process development for antibodies and viral vectors. So in 2022, I moved back to China with my family and joined GenScript Probio to lead our CMC platform and for next generation biologics like bispecific antibodies, fusion proteins, ADCs, and more. So it's been an exciting transition working at the intersection of science and manufacturing to bring complex biologics from concept to clinic and to commercialization. I'm thrilled to be here and look forward to sharing the insights with you today. Thank you.
Thank you for sharing your personal story, Ying. Now let's take a closer look at Probio itself, and let's start with the short video you brought today.
From concept to cure, we are your global CDMO partner in biologics and cell and gene therapy. Let's shape the future of medicine together. With every technological advancement, the journey of scientific discovery continues. At Probio, we stand with our partners, transforming breakthroughs into real treatments that change lives. With integrity, innovation, and a commitment to quality, we accelerate the future of medicine, bringing therapies from concept to cure. Probio, customized CDMO solutions.
Great, nice introduction, Ying. Now I would like to ask you, you have prepared some presentation slides to introduce you to the company, please.
All right. I hope you can see my screens.
Yes, all fine.
Okay, perfect. Yeah. All right, so it's my pleasure to introduce the Probio Edge, powering next generation biologics with CMC excellence. So first and foremost, a little bit background about Probio. Our vision is to be a leading global CDMO, renowned for cutting-edge technology to enable our customers to deliver life-changing therapies worldwide. And our mission is through our commitment to quality, collaboration, and innovation to deliver customized solutions to transform scientific discoveries into market-ready therapies and to make a lasting impact on patients' health and well-being. And our values, core values, include customer-centric innovation and integrity. So actually, Probio is a subsidiary company of GenScript Corporation. So just a little bit background on GenScript and our strategic positioning within the corporation. So GenScript was initially founded in 2002 in New Jersey, US, and quickly becomes the world well-known provider for gene synthesis. And this is known for their life science services and products. And later, we have the Biosyn, which is focusing on the industrial synthetic biology products. And Legend Biotech, which is also located in the US, which provide cutting-edge cell therapy products, the CAR-T cell therapy. Over the years, it collaborates with actually Johnson & Johnson to launch one of the most successful CAR-T cell therapy product called CARVYKTI, targeting for multiple myeloma, and which was being commercialized in 2024 in the US and later across the globe in a number of different countries. And last but not least, of course, Probio, which is the biologic CDMO franchise under GenScript. So our journey of rapid growth for Probio actually dates back to about 2 decades ago. From 2008 to 2018, we were actually part as a biologics department under the GenScript Life Science Group. And we have obtained our first IND in 2017 with a complex recombinant protein case. So in 2019 and 2020, Probio was basically launched as a separate company, and the Probio brand was established. Also, the Nanjing Biologics R&D Center, as well as our GMP manufacturing center, was put into operation in Nanjing, China. In 2021 through 2023, we received a series of funding from the well-known investors in China. With this increasing capitals and investments, it enables us to increase our capacities and capabilities across the globe. In 2021 and 2022, during the COVID period, we successfully supported a number of our partners to launch their COVID vaccine and COVID antibody products for emergency use authorization. And in 2023, we signed our first BLA and commercialization CMO case with a Shuyang pharmaceutical company in China. And last year in 2024, we observed our ground launch of GMP biologics and LVV commercial production site in Zhenjiang, China, as well as the new addition of our Hopewell site at New Jersey in the US. So this slide shows a global footprint of Probio's platforms. If we take a look at the biologics side, we have 3 major locations in China, Nanjing, which is marked as number 1. That include our discovery site and discovery lab, as well as biologics process development, cell line development, as well as clinical stage manufacturing facilities. In Zhenjiang site, which is marked as number 2, which hosted our biologics process development, as well as commercial and large-scale GMP manufacturing facilities. And number 3 in the US, Hopewell site, for biologics, we have currently PD-AD lab as well as GMP manufacturing facilities under planned. And on top of MABs, we also have a lot of capabilities to host other modalities such as virus, plasmid, and mRNA across the globe. With respect to the track record of different modalities, for biologics, we have obtained over 16 international license out projects, over 100 international CMC and contract manufacturing organization projects, over 50 global IND approvals, and 170 GMP batches completed. And for cell and gene therapy products, over 60 global CMC projects and 70 IND approvals from NMPA, FDA, and other regulatory agencies across the world for different types of cell therapy and gene therapy products. If we take a deep dive into the scope of biologics CDMO business, we cover early phase discovery, including state-of-art, pro-speed, single B-cell platform, fully human antibody discovery and antibody engineering, as well as our SMAP bispecific antibody platform. This is our proprietary platform, offered a unique design of antibody plus single domain antibody for a bispecific antibody format. Moving into preclinical development, we offer proprietary pro-cell line development services, as well as end-to-end upstream, downstream analytical and formulation development. For early stage clinical manufacturing, we have 200 to 500 liter scale clinical manufacturing capabilities in our Nanjing site, as well as compatible pilot scale fill and finish covering a variety of different dosage forms, such as liquid, lyophilized powders, and pre-filled syringe. For late stage clinical and commercialization capabilities, we conduct process characterization, process validation studies, as well as 2,000 liter scale commercial manufacturing and commercial scale fill and finish. We are actually very proud in our expertise in developing complex molecules, including different type of multi-specific antibodies as well as proteins. So roughly speaking, about 50% of the projects that's been delivered for IND approvals are proteins and bispecific antibodies, and the rest of 50 are monoclonal antibody molecules. So here is a library of different molecules examples that has been successfully delivered by our platform. With respect to bispecific antibody, they can be categorized into either IgG-like, which contains the Fc fragment, and non-IgG-like, which contains the Fab fragment. So we have done symmetric bispecific antibody as well as asymmetric bispecific antibody. For example, the IgG plus single chain Fv fragment, IgG plus VHH single domain antibody, or IgG plus peptides as well. For asymmetric bispecific antibody, we have done cases with asymmetric ones with 2, 3, or 4 different chains, as well as IgG plus cytokines such as interleukin, as well as trispecific and multispecific antibody cases. Moving on to the Fab-related molecules, we have completed a number of cases, and we have seen increasing popularities in antibody fragments with in vivo CAR T therapies, such as Fab, single chain Fv, as well as VHH, and a few cases with the conjugates with different antibody fragments, such as single chain Fv conjugation in vitro, single chain Fv plus Fab, and different fragments in conjugation with LNP molecule. For the protein type, there are fusion proteins such as Fab fusion protein as well as Fc fusion proteins. And for a few cases for recombinant proteins that covers the trimer vaccines and hormone cases and unstable enzyme molecule as well as blood factor 8. And all these molecules pose quite unique challenges in terms of CMC and manufacturing, which we have successfully tackled those challenges and established our platform solution. We have summarized these lessons learned from over 100 CMC projects and over 200 challenges and solutions in process development into our intelligence database called ProBox. And currently we are basically incorporating AI functions into our database to create the ChatGPT version of process development based on our own database and intelligence. And here we offer quite flexible services as well, from small to large scale GMP manufacturing and process development capability to support different modalities as listed here. And also We have done a number of cases, perfusion and intensified fed batch mode for upstream. So we have established the perfusion system to support up to 300-liter perfusion batch. And for pilot and commercial scale, aseptic filling line with multiple different formulations, such as pre-filled syringe and nasal spray. These are quite unconventional dosage forms, and with a variety of different vials volume for liquid and lyophilized powder solutions. So this is my last slide. So I'd like to highlight that so over the past few years, we have seen BD licensing and other forms of global collaboration have become a growing trend in the biopharmaceutical industry. So our company has successfully supported our partners in a number of BD transactions, including 6 license out projects as well as 1 acquisition projects, with a complete list shown here. So a couple of highlights. Over the last year, we supported 2 Chinese local biotech companies to complete their license out deal with global MNCs. And their products are actually, one of them is a tri-specific antibody molecule, and the other one a bispecific antibody. So through our study, we provide pre-IND and IND enabling CMC services and help to perform the due diligence and audits from global pharmaceutical companies. With that concludes my presentation about our company. So happy to move on to the question and answer session. Thank you.
Thank you very much, Ying, for this overview. And to the attendees, if you would like to ask any questions, please type them in the chat and I will ask them later at the end of the interview. But of course, I have prepared some questions for you, Ying. First question would be, so what differentiates Probio from other biologics CDMOs? What would you answer on that? What USP can you give us?
So I would say there are a few key features that set us apart from other CDMO. First, Probio offers end-to-end biologics CDMO services, truly from the early phase antibody discovery and engineering to stable cell line development, process development, analytical development, and commercial and clinical phase GMP manufacturing, as you can probably see from my presentation. And this seamless DNA, I think GenScript could also support with us with the gen synthesis service as well. So this seamless truly DNA to GMP platform is underpinned by our deep expertise in both antibody discovery as well as CMC. So on top of that, we also offer comprehensive services covering a variety of different modalities on top of biologics, different very complex formats of proteins and antibodies. We also do antibody drug conjugates as well as cell and gene therapy, plasmids, viral vectors, and mRNA, which enable us to provide one-stop solution for some complex modalities, such as if you need conjugation with antibody or, for example, with mRNA, LNP, we could do one-stop solution for you. And the third point is our global reach and local flexibility. We actually operate on a global scale with our lab and manufacturing facilities in both China and the US. That meets the global regulatory standards. We also have our local offices in Europe, which is based in the Netherlands and South Korea, Singapore, etc., to support local customers across the globe. Last but not least, I'd like to highlight our exceptional fast timelines. So from gen synthesis to tox batch release, we can conduct these studies within just 6 months for standard monoclonal antibodies, and as fast as 7 months for bispecific antibodies.
Thank you very much. So let's dig a bit deeper into the science. Could you give us some insights into your innovative scientific approach? As you mentioned already, the Probox toolbox for CMC, for example.
Yeah, absolutely. Innovation is truly at the heart of what we do. We do have actually a dedicated R&D team working on platform innovation part. And over the years, we have built a science-driven platform that supports a variety range of modalities as well as challenges. So a couple of examples I'd like to highlight. So first, our proprietary cell line and expression system, and they are engineered for high productivity and stability, and to ensure robust upstream performance across different biologics. And also over the past few years, we have also developed cell lines that can enable different certain glycosylation patterns, such as our ADCC plus cell line with afucosylation antibody production. And we also recently generated our cell line that could generate high cellulation level to enable to enable longer half-life for the antibody. And
Okay.
In addition, we also developed our own chemically defined animal component-free cell culture medium in-house, and we are now collaborating with Thermo Fisher to manufacture this in-house media for GMP production. So with our own in-house media, we can successfully reduce the cost by over 90%. And on top of that, our own medium is quite compatible with our own cell line and expression system, which through testing in a number of IND projects, we have seen our own medium stand out as the highest titer and good quality performance in roughly 70% of our projects. And second, as you mentioned, I think I mentioned in my presentation as well, our toolbox platform, which is our knowledge-driven process development toolbox to help us troubleshooting and enable faster and more reliable process development process. And for complex modalities such as ADCs and radioconjugates, our AXC conjugation platform provides precise and scalable conjugation strategies covering random conjugation as well as site-specific conjugation, ensuring high product quality and consistency. So together, I believe these innovations enable faster cell development timeline and greater molecular developability and smoother regulatory pathways for our collaborators.
Thank you very much. So, and how does your platform process evolve for complex biologics like bispecific antibodies, fusion proteins, or ADCs?
Yeah, so this conventional so-called template process or platform process is for conventional mAb molecules. And as biologics became more complex, and our platform process have also evolved significantly to meet their unique development and manufacturing needs. For bispecific antibodies, we've optimized our expression systems and upstream strategies to manage the chain parents for asymmetric bispecific antibodies and enhance the yield. In downstream, we developed tailored purification approaches to efficiently remove the mispaired species and aggregates. Those are typically key challenges for bispecific antibody molecules. And for fusion proteins, often they come with stability challenges. So we adjust our cell culture conditions and formulation strategies accordingly. And also for those proteins without the FC fractions, we've designed our platform capture process for those type of proteins that do not contain the FC fragment with good overall capacity and purity upon capture step. And for ADCs, we optimize the conjugation process to ensure the high efficiency in terms of conjugation reaction with well-controlled DAR and ensure its successful scale up from lab scale of 1 to 2 milliliter up to like a few hundreds and tens of liters in the conjugation reaction. And also design corresponding analytical platform to characterize the critical quality attributes of this product. So in summary, the new platform process is not actually built upfront as we gained a lot of experience and knowledge from working on these complex molecule projects, we learn their unique problems and identified effective solutions to address these problems, and therefore gradually customize our conventional template process to meet the needs for new and challenging modalities.
Okay, thank you very much. So we're running a bit out of time, but I still would like to ask you if you could give one example for a successfully delivered CMC.
Of course. So I'll give the example for the coagulation factor 8, which is a quite complex molecule with very low yield, which is prone to form aggregates and easy to form fragments. It has complex PTM, and we also need to address the stability study through the formulation and come up with a good bioanalytical development to support the release. And so we delivered IND enabling CMC services and support our client to obtain IND clearance in 2022. And this molecule is currently in clinical phase 3 in process. And we help the client to do late phase process optimization, which will help reduce the cost of goods by 60% upon optimization. And currently we are supporting the client to file BLA very soon, hopefully by the end of this year.
Great. So we already have some questions from attendees, but before we start with them, I would like to know you're part of a strong innovation network. So how can you help other members of bioRN or the broader biotech community?
Yeah, so we're always here ready to support our partners, which include biotechs, biopharmers, and our academic groups. So we can do this through two major mechanisms. First, as an end-to-end CDMO, I think we learn pretty much about our company. So we support our partners throughout the entire journey from early phase drug development to CMC studies, to preclinical and clinical manufacturing, all the way to commercial production. And the second way is we also collaborate with our partners to accelerate innovation through platform technology licensing, which include, for example, our proprietary cell line and vector system, our SMAP bispecific antibody platform to enable much faster hit generation and minimal engineering, and also our NME, which is called new molecular entity, which include over 30 pre-development MAP bispecific antibody and single domain antibody assets to readily enable a new molecule development for different modalities such as TCE, ADC, CAR-T, etc.
Thank you. So let's start with the attendees question. First comes from Daria. What kind of manufacturing setup do you use for your CGT modality? Do you use closed system, modular building, robotic solutions to handle biologic materials?
That's a very good question. However, I'm from the biologic side, so I don't know if I can address this question very accurately for the CGT side. I think I will leave these questions to our CGT team, and we will get back to you after this session.
Great. Okay, so the next question is, what methods do you use to identify best antibodies, single B cell platform?
Very good. For single B, I think this question belongs to, belongs to our discovery platform. Again, I come from the biologic CMC side. Methods to use. So I don't know exactly what methods to identify best antibodies. So good question again. I think I will ask our discovery colleagues to get back to you after the session.
Great, Pavel and Daria, we will come back with the answers. So next question from Friedemann. For your technological developments, do you draw on external innovation? Do you license external tech or buy innovative startups, etc.?
Yeah, I think I sort of mentioned from last question. So we do license out our own technologies, and at the same time, we work closely with some companies to do licensing as well. A good example is for our ADC discovery. I think we license in some the site-specific conjugation with based on glycosylation conjugation. So that particular technology is we work with our external partners for licensing technology, and also currently with the emergency power of AI, we are partnering with a number of AI companies to speed up our early phase discovery. And also for our CMC studies, we're looking for some external partners to collaborate our CMC process development, including digital twin models, as well as looking for good drug developability screenings using AI, as well as potentially for early phase formulation screening as well.
Great. And last question comes again from Pavel. What kind of conjugation chemistry do you mostly use, for example, for the SCFV PEG-based conjugates?
Yeah. So we mostly, currently, I would see at this stage, mostly we still work with lysine-based or cysteine-based conjugation. For example, for this SCFV PEG-based conjugates, this is involves actually a 4-step conjugation. So first, we manufacture 2 separate SCFV molecules, and we conjugate, which both contains C-terminal cysteine. And we conjugate a linker through the cysteine maleimide conjugation mechanism on the cysteine side. And on the other side, we have another attachment through click chemistry, I think, with the DBCO group with the N3 click chemistry mechanism. So actually, this is the PEG molecule is incorporated in both of the linkers with a purpose to extend the stability and half-life of the single trans-weight as well as conjugates.
Great, thank you. We're almost at the end, but I would like to ask you one question. If you could share one piece of advice for young biotech companies trying to bring a complex biologic to market faster and more reliably, what would it be?
Yeah, that's a great question. So I would say, of course, focusing on the science, and of course, balance the science and potential success. Because we work with a lot of startup companies, and a lot of them working on cutting edge technologies, and which include quite fancy technologies, especially in some emerging modalities, such as the antibody conjugates, as well as the cell therapy, for example, the in vivo CAR T therapy, etc. So first of all, always do good science, focusing on the cutting edge technologies and working with good partners like Probio to help you overcome some of your challenges in your early phase development. And also, I would say do not overlook the importance of, for example, the developability of the molecule and potential chance of moving forward into the late phase. Otherwise, sometimes if the molecule of design is actually not suitable to be developed to moving into clinical phase or into later phase commercialization, that could be also one of the major hurdles to help to put these molecules moving forward and being successful. Yeah.
Thank you very much. I see there are 2 other questions come in the chat. I will forward them to you because we're really running out of time. Thank you everyone in the audience for joining us today. The next member-to-member session will take place October 9th, and we are going to interview M2M. So thank you very much. Appreciate having you here today. Have a nice time. Have a wonderful summer. Thank you and bye-bye.
Thank you all.
Thank you.
Bye-bye.
Bye-bye.